Good morning, Pawan sir.
Yeah, very good morning. Can you hear me?
Yes, sir.
Okay. Can the screen be enabled? Just we tested the slides are working.
Yes. We shared the host, sir.
Okay. Is it my screen visible?
Yes, sir.
So is it in full screen or as a presentation?
It's in a presentation.
Okay. Now?
Yes, sir. It's full screen.
Yeah, okay. Okay. You can also hear me perfectly, right?
Yes, sir.
Okay. Perfect. Slides moving, right?
Yes, sir.
Okay. Okay.
Good morning, Tiwari, sir.
Good morning, madam. Dr. Pawan, good morning.
Sir, very good morning. How are you, sir?
I am fine. We
Okay.
all
Are
have
you?
left. We all have left LG for you.
Okay. Okay. Thank you.
We have not spoken anything during last 5 days on the LG.
Okay. 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 Okay.
So you are free to cover in 1 hour.
Yeah. Yeah. Sure. Sure. I'll
So
try to make
LG
it.
session for you. We have
Okay. Thank you. Yeah.
given walkover to you.
Okay. Sure. Sure.
We are going to start soon.
Yeah. Yeah. I'm ready. Fine.
Good morning, everyone. A warm welcome to the fifth day session of Faculty Development Program on Frontiers in Life Sciences and Value-Added Products from Bioresources, conducted by the Department of Biotechnology, VFSTR, in collaboration with Indian Agricultural Research Institute. Today promises to be yet another insightful session filled with valuable knowledge and engaging discussion by our speaker, Dr. Pawan Juttur, on the topic algal farming, a sustainable triad for bioresource innovation. It's time to introduce our today's guest, Dr. Pawan Juttur.
Good morning, all. It's an honor to introduce Dr. Panaga Pawan Juttur, PhD. Good morning, one and all. It's an honor to introduce Dr. Panaga Pawan Juttur, PhD, a distinguished researcher and group leader at the International Center for Genetic Engineering and Biotechnology in New Delhi.
Dr. Jutter's expertise lies in algal biology, where his research is dedicated to understanding microalgal systems and developing strategies for sustainable biofuel production. With academic credentials from Pondicherry University and postdoctoral experience at the University of Nebraska, Lincoln, USA, Dr. Jutter brings a wealth of knowledge to his field. His work focuses on reconstructing metabolic models to convert light and carbon sources into biomass and biofuels, addressing concerns over fossil fuel depletion and global warming. Microalgae under Dr. Jutter's study holds immense promise as feedstock for renewable biofuels due to their unique ability to convert atmospheric CO2 into valuable resources. His research delves into an intricate biology and metabolic pathways of microalgae, particularly in fatty acids and triacylglyceric synthesis under varying environmental conditions. Dr. Jetter's dedication to unraveling biological systems and identifying key pathways for biofuel production is evident through his integrated omics research. His contributions reflected in numerous research papers highlights his significant role in advancing algal biotechnology. Beyond his research, Dr. Jeter actively engages in professional societies and advocates for further exploration microalgae genetic and metabolic engineering. His commitment to advancing biotechnological approaches for sustainable energy solutions earns his respect within the scientific community. Please join me in welcoming Dr. Pannaga Pamanjitur, whose pioneering research continues to drive innovation and sustainability in algal biotechnology. Thank you. Sir, the forum is
Yeah.
all yours. We are sharing the host access to you. Sir, please.
Thank you very much for that kind introduction and little brief notes on what we work in that. So very good morning to all the participants. And first of all, I should thank the organizers for inviting me as a resource person for this e-FDP event, which is entitled as Frontiers in Life Sciences and Value-Added Products from Bioresources. So it's my great opportunity to discuss what kind of work we are working at ICGB, and also share what basic research and parallelly translational research approach can help us in exploring the sustainable triad for bioresource innovation. So today's topic, I'll be discussing an alga farming and how we can look upon these different kinds of this enriched source of bioresources for understanding in nature how they behave in different conditions. So to start, to start with, let me introduce where I'm working on. And as everybody knows that ICGB is a UNIDO organization, UN-based organization, which involves in different programs like not only research, and also we have some fellowship programs. We also do conduct some meetings, grant applications, and also we are also involved in technology transfer. Onto my center part, you can see the core areas of ICGB where we are working on infectious diseases, non-communicable diseases, medical biotechnology, industrial biotechnology, and plant biology and biotechnology, where I am currently working on biofuel and renewable energy as its third generation biofuel, especially the model system as algae. And also we are trying to understand different scenarios of algae through basic research to fundamental research. So what are our strategic plan of ICGB is we are also, as that, we are a leading intergovernmental organization involved in research, training, and technology transfer in the field of life sciences. And our mission is to contribute scientific research with the capacity enhancement, thereby promoting sustainable goal development. So ICGB is involved in the core areas like zero hunger, promoting health and well-being for all, quality education for all, promote access to affordable and sustainable energy, promote innovation, and also global partnership for sustainable goals. Along with that, we are also involved with cross-cutting actions with the SDG 5, 12, 13, and 15. That's how our strategic involvement of research in different groups work upon. And we have several interdisciplinary areas where we work on different concepts of microalgae, second generation biofuels. Not only that, we also work on plant biotechnology, where we work on different aspects of environmental stress and also promoting health and well-being, that is designing of vaccines and different kits for infectious disease and all. So we were lucky enough during COVID time, our institute, some of our colleagues were involved with COVID research in terms of understanding COVID in different scenarios. So today for the talk, because we have to talk about uh, algal farming as a basic this thing, because I don't want to go for introduction. What is the basic algae? We know algae as a different forms. We have cyanobacteria, microalgae, and macroalgae, which is unique, and they have their unique characteristic of how they are sustainable and they can produce different molecules which are relevant for the industrial biotechnology. So we have to explore the circular biomanufacturing through harvesting different concepts. So today we are going to talk about, in my introduction also, we are working on carbon sequestration, which is the need of our, because environmental conditions and environmental CO2 sequestration is very important and need of the hour. The only source where we can work upon those is microalgae and plant systems, where you can sequester CO2 in different high value products. And you can see from this overall graph, where we can see that you can capture sunlight and you can convert in form of solar energy, and also you can generate electricity and also can be used as a biofuels. And mainly today's talk, we are going to talk about something on a basic research, how algal factories can be used in sequestering CO2 for production of various high value products, naming from vitamins, vanillins, or any polyhydroxy and resveratrol. These are all very high commercial products which can be used as a bulk products, which can be used in day-to-day. And also we are applying a strategy of circular biomanufacturing. That means the net energy out is almost net zero. So that's how we can work upon, and that's a need of our, like later in the future, how we can look upon and also control the different patterns of that, where you can sequester carbon dioxide, you can clean up waste water, and also use basic resources which are available, and we can convert them into high value products which are useful in different products what we use. That's how we can add a greater value in terms of that. And we are today exclusively discussing about how algal biofactories can be used as a precursors for producing different bioresources. To understand that concept, let me highlight our roadmap and what we are working in our group. This is almost the PhD consolidated summary of 7 PhDs who have been just graduated last year is from my group. And what we try to put upon is, this is the overall roadmap, how you can achieve different molecules. And to understand that, you need to understand the basic biology, and understanding the basic biology, you can translate that from the lab to the industry, where you can apply these technologies in a form of algal biorefineries, where sustainable production of biofuels can be substituted by production of high value products. So I'll be taking a little bit time on this roadmap, because this is very important for all of us to understand and also take care of that. So basically what we do in the lab, if I go to my left side, it's more of not even genetic engineering. We look upon a non-GMO approach, where no need of modifying these organisms to produce any kind of these products, because in nature these products these algae are capable of producing multiple multiple products where they are capable of producing these very relevant high value products. So we have to understand how in the environment these microalgae are working to sustain these processes, and they're able to convert different energy resources, especially light, nutrients, and the water, using those three basic components into three different components, that is biomass, biofuels, and biorenewables. That's why we call it as a concept of B3, where we are trying to understand microalgal systems through integrated omics approach to enhance the production of biomass, biofuels, and biorenewables. So these are the net products which we are going to obtain, whatever concept we are trying to understand upon. What exactly we look upon? Okay, so there are different types of strains. For example, there are microalgal strains. Especially we are talking about microalgae, not on cyanobacteria and macroalgae. There are different concepts what cyanobacteria can produce, what microalgae can produce, and macroalgae produce. To go for a nutshell, cyanobacteria are able to produce not only proteins, that also can produce at PHBs, which is polyhydroxybutyrates, which are very important sources. Whereas macroalgae like seaweeds, which are more popular as a food ingredients, they are rich in carbohydrates, so they can be an alternative source for the fourth generation biofuels, where we cannot get lignocellulosic biomass or anything. So because the major hurdle of lignocellulosic biomass can be overstood because macroalgae don't have lignin part. So in that way, lignin is not available, and it is very easy to extract the sugars from the macroalgae, so that sugars can be fermented into second generation bioethanol and any other products. So macroalgae is a future sustainable feedstock for production of kind of biodiesel and all. So this is one area which has to be explored at basic level, so that in order to understand those and also to capture the how, and also not only that, macroalgae is able to help to sequester a lot of carbon dioxide from the environment. That is where we see in oceans huge algal blooms, which are also forming as a form of islands, which can be a huge opportunity for carbon sequestration also. So coming to the overall research perspectives, what we work upon. So we have multiple strains, and remember friends, like usually one microalgae cannot produce multiple products. The basic components of any microalgae, it may produce a carbohydrate, it may produce a lipid, or along with that, it can produce carotenoids. What are these carotenoids? We are going to look upon that, and that is the core area, because that is where we can substitute these carotenoids so that bring the value of biofuels down. So what we are going to look upon, first step, what we are going to look upon is how to engineer your strain. So basically, we call the concept of media engineering, so that you can select a strain of your interest from the nature or a repository and do different media engineering. What is media engineering? Media engineering is a concept where you can manipulate or modify your basic media components like nitrogen, phosphate, sulfur, along with substitution of some other compounds, so that you can produce relevant products. Okay. When we see the phototrophy conditions, basically they need a light, nutrients, and some kind of a modulators or water sources to convert into any product that is a lipid or high value products such as carotenoids or any other vitamins. So we have a concept where we try to understand how different chemical modulators can modulate your microalgae to produce those products, or how these nutrients can help in same thing, light and carbon dioxide are the major role, because it's a phototrophic microalgae. So in that scenario, also the new concept what we are trying to look upon, it is not a new concept, but we are trying to look upon how in the nature these microalgae consortium are able to produce very good relevant products and that concept is called co-cultivation, where today's talk will be based on that, how we can produce using naturally symbiotic bacteria or any other organism with algae, how they can enhance the high value products within the microalgae system. So mixotrophy is again the combination where you use some kind of a sugar substrate, either in dark or light, or you substitute with other substance, and basically you try to combine the heterotrophy and phototrophy in order to produce. So microalgae can be either produced in a form of heterotrophy or phototrophy or mixotrophy together. So heterotrophy, there are many products which are produced, which is I'm going to talk in a minute. So where you can convert the sugar sugar molecules or simple byproducts of biorefineries, which are crude glycerol, into high value products like omega-3 fatty acids. So that is one classical example where people want an alternative omegas from not only from basically the omegas are from fish, we know that. So we need an alternative source of omegas, which is vegan omegas, which is DHA and EPA, and these can be alternatively producing using algae, and that is how you can use alternative sources like crude glycerol, because sugar will not be available. Most of the sugar will be used for fermentation, either by ethanol or alcohol companies, because sugar will not be a substrate for those things. So we also developed a technology where other than simple glucose or other sugar molecules, we can utilize the byproducts like glycerol, the waste crude glycerol, which is a byproduct of biofuel refinery, can be used to convert that glycerol into high value products such as omega-3 fatty acids. So this mode does not require light as an energy source. So we do in a heterotrophy mode where we substitute using glucose or any other equivalent to glucose molecules in order to produce high value products like DHA and EPA. So this is how heterotrophy also plays a major role in converting the byproducts, that is waste to wealth kind of thing. So most of our concepts is how do you use minimal sources and available sources such as waste, maybe carbon dioxide, which is there in the environment, which is by sequestering the flue gas, or using the wastewater as a media source, because algae needs a wastewater as a very important source for that, and also use these byproducts from the refineries where these products are of no use, can be reused by as a substrate for the algae and convert them into high value products. Thus, by selecting the strains, you can engineer those strains using media engineering approach in order to understand the basic biology of that. So in our group, what we do is when you come to the center, this part 2 of our roadmap, mainly it consists of integrated omics, what is also known as multi-omics approach, what we call it as iomics. So basically, what is iomics? So iomics is a new tool or a molecular tool to understand the whole micro behavior of the cell at a physiological level, what is happening at the metabolic pathway level. So basically, we can do via forward omics approach or reverse omics approach in order to understand this. Forward, I'll just brief what is forward omics approach. So if you know the sequence of the particular strain, we start from genomics approach, then do some transcriptomics, proteomics, and metabolomics, and try to integrate that data in order to understand how the cell is behaving at a particular approach, at a particular environmental constraints. So what is reverse omics? Reverse omics is the opposite to that, where if we don't have a genome of a particular strain, because we have thousands of microalgal strains, all the genomes are not available. Till date, we have maybe around 100, 150 good annotated genomes which are available in the microalgal repositories. And these are enough to understand the other microalgal systems, because still even the cost of the omics, like platforms like Illumina sequencing or PacBio sequencing has come down. The sequencing approach is still expensive in terms of doing many kinds of full sequence analysis. So what In order to quickly understand the concept, we will try to go through reverse omics approach, which we start from understanding the metabolic profiles of that pathway during those interactions. It may be a nutrient stress interaction, it may be carbon sequestration interaction, it may be a co-cultivation interaction. Without knowing the uh, genetic makeup of an organism, that is genes or transcripts or proteins, we can able to hypothesize how the cells are behaving in a particular environmental condition. So this is a very new approach where you can go ahead with understanding from the metabolomics part initially, and then map it on to different pathways based on the, because metabolites don't change. The ultimate metabolites, even it is in E. coli, it may be in human, it may be in anything, metabolites are same. So for example, glucose is same component in all the things. So thus the metabolites or metabolite constituents does not change in whatever course you're trying to do. So what we try to do is we quantify those metabolites or qualitatively analyze those metabolites. What are the metabolites which are part, because metabolites are the main molecules which are interacting with each other so that it can produce it can produce a product of our interest. So understanding the metabolomics is very important, and also that metabolomics can also go further in order to understand through proteomics and transcriptomics, and that can be overlaid to understand the overall mechanism. That's how our basic research, it's very important. When you want to make it a GMO approach, that is genetically modified organism, you have to understand how the cell is behaving. There are two concepts. Either you can introduce a foreign gene into the cells of microalgae and overexpress the product of our interest. And second concept is you understand the basic biology through integrated omics, the whole cell at a time, and that will give you a clear picture what are the pathways which you can shut down so that your carbon flux will go towards high value products or biofuels which you are interested in. So in our lab, we also do a work called transcriptional engineering, where we identify through omics different transcriptional factors and see whether these transcription factors are upregulating or downregulating the functionality of these transcription factors, so that if you shut down those transcription factors, though the carbon flux will be diverted towards the molecules of your interest. For example, one of the BIRAG project, where we worked is we identified a transcription factor called BZIP, which is involved in increasing the lipid content in microalgae. So what we have done is we have shut down that BZIP transcription factor using CRISPR, RNP-based CRISPR technology, so that the carbon flux can go towards production of astaxanthin. Thus, we were successful and demonstrated that your carbon can be diverted instead of going for lipids. Once we shut down that BZIP, which is involved in that pathway, the carbon flux goes towards the production of carotenoids such as astaxanthin. So this is a new concept of transcriptional engineering, which is upcoming, where you no need to disturb the overall pathway. That means you no need to manipulate one gene at a time, but you can understand what is the transcriptional regulator or transcriptional gene which is regulating the pathway biosynthesis, and shut down or overexpress these genes so that you can produce the products of your interest. But a very important scenario is you have to maintain the carbon and nitrogen balance, because microalgal, most of the products are formed based on carbon and nitrogen balance. So in that way, we try to understand the overall insights of molecular mechanisms, what is happening with different equations or environmental factors such as CO2 sequestration or co-cultivation or nutrient stress. Nutrient stress, people who are working on algal research know that nutrient stress is a well executed phenomena, but we always end up in compromising the biomass. So our lab or our group tries to understand the scenario where you no need to compromise your biomass, but also produce the biofuels or high value products of your interest. So in that scenario, on the center part of the step 3, what we are trying to look on 3 components, that is biomass. That is, how can we enhance the biomass to a level such that it can be used for production of your relevant molecule. So biomass is a very important criteria algae, because it is better than other organisms to grow upon, but you need to work upon biomass in a quicker way so that it can produce a product of your interest. So we have worked on certain projects where we work extensively on understanding the biomass, where we sequester with CO2 and enhance the growth. So for example, in a layman terms, what I can say, if a cell is growing, if a doubling time of a cell is around 4 days, using carbon sequestration and nitrogen balance, Ca by N ratio, we try to cut down the doubling time to 2 days or 1 day. So this is how you can increase the biomass, not only by reducing the doubling time, but also produce the product in a quicker way. So that is one concept you have to understand. What is the reason why you are not getting enough biomass, and how you can produce enough biomass so that you can produce enough product. Because once your biomass is increasing, the biomolecules like as lipids or any high value products within, that is carotenoids, vitamins, also increases depending on what strain you are using. So in that concept, we try to understand different microalgae where you can produce enough biomass so that that can be procured for producing this thing. The second problem where we are looking upon is production of biofuels. So biofuels are very important. At this point, we need fine tuning of biofuel production, especially from natural resources. And this third generation biofuels are equally important in order to produce. And why they are important? Because by 2050, if the fossil fuels get depleted, we need an alternative source for our biofuels, and that is where we are working upon. And so basically producing lipid molecules, jet-based biofuels in the microalgae is very important, and that's how we try to work upon oleaginous microalgae, which can produce lipid precursors, which can be used as a biodiesel and also as a jet fuel molecules. The other important thing what we are talking about in terms of our today's FDP is biorenewables. So biorenewables are very important. We know that biofuels are not sustainable at this point of view. So we have to have a alternative so that it can become sustainable and cost effective. So substituting, as I said in my initial comment, that algae produces one product. Along with that, they have an alternative product, that is co-production of high value products. You have to capture what kind of products can be produced by algae, and that thus enhance the product of that interest. So now, along with the lipid, there are some molecules which can be produced. Those are vitamins, or such as tocopherols, or it may be a fucoxanthin or zeaxanthin or lutein or astaxanthin in that case of transcriptional engineering, what I was talking about. So there are multiple products which are being produced, and these are produced in minimum quantities, which are very important for pharmaceutical or nutraceutical approach. So producing these products which are high value can bring down the cost of the biofuels. So that is one alternative way. If you can co-extract your high value products initially from the products, come to the algae, and use the remaining biomass for production of lipids or production of extraction of lipids by supercritical or HTL process, that will bring down the overall cost of your biofuel production, which should be around $100 to $150 per gallon. So this is what the trademark we have to apply upon, and we have to work it out so that it will be competitive to produce the fossil fuels. This is why biorefineries concept is always very, very promising in terms of not only biofuels, you have to produce high value products so that the overall cost will reduce. That's why we call it as a roadmap end to end. So substituting, understanding, first thing is basic research of how microalgae is behaving, and capture those things for production of high value products and lipids of what you saw. So overall biorefinery is that such kind of a roadmap where you should have multi-product or multifaceted applications of those different products, so that you can use for substituting these products, and overall cost can be. That's how you can make cost effective and sustainable biofuels or biorenewables. So this is what our group works upon, and this is how, because I took good time on this to explain what we work upon, and today I'm going to um, talk about one very important thing, because this is a need of our, because we have to learn science from observing the environment. So basically, we want to understand how in an environment a microalgae or a bacteria can interact with each other so that it benefits the microalgae for production of the value added products. So the concept is, how do we elucidate the functional interactions among algal microbiomes? And the whole concept we are trying to understand by using the concept of reverse omics. That means we'll be applying metabolomics, and this is like, for example, these two strains which were identified or which we worked upon were not sequenced. That's why we thought, like, let us go with reverse omics approach to understand what is the functional interactions among these microbiomes, so that it will help us to further illustrate how these microorganisms are working together in the environment, so that that can capture the value assets what we are talking about. Basically, microalgae or any bacteria within the in the natural environment is always coexist as a consortium or a microbiome. So this is very important to understand how the mechanisms work upon, and we try to understand through on to my right, this is my PhD student from Tanzania who is working on this project, and he is going to submit in next 6 months. Almost done his work. So I'm going to present how we have demonstrated the interactions between a microbe and bacteria, so that the consortium will help both bacteria and microbe to interact with each other, so that that coexistence will help us in order to produce high value products of our interest. So the case study what we are going to discuss, this is an example, very classical example, which has been published last year, where they have seen that in the natural environment or a lifestyle, there is a lifestyle switch between a bacteria called sulfitobacteria and an algae called Emmelinia huxley. So these basically have produced certain products. For example, you can see onto my left of this figure, where my pointer is showing, like products like DMSP, benzoate, which are produced by the bacteria or taken up by the algae, and the algae also produces some products which are taken up by bacteria for the coexistence, so that they'll not cause any harmfulness. Same thing when there are certain products which are produced by bacteria which are virulent, can cause, and the bacteria can use the growth substrate from the algae to grow up, and that can lead to the pathogenicity of the system. So basically what I'm talking about pathogenicity is, this is how the algal blooms come upon. So there are the interactions which are happening in nature, where it may be a pathogenicity or coexistence depending on the conditions you apply upon. So it is important to understand whether such factors are leading to coexistence or pathogenicity of the algae. It is very important to understand, so that we can capture the coexistence phenomena through integrated omics approach in order to fulfill the production of required product. So onto my left, you can see a lake called Lake Natron. So you can go to Google and just put as a Natron Flamingo Lake. It is a very famous lake in Tanzania, where the pond, what we call, it's a big lake. Okay, so it's a big lake where it has different colors during a season. So this is a capture of a capture from the where they can show different colors, and you can see that the microbial chemical composition is being showed. So deep blue in between is pure water. Okay. And the green color is the chlorophyll, that is nothing but microalgae or algae. The orange yellow on the edges or red color is microbial carotenoids. So these carotenoids are produced by the microalgae or are left into this thing. And the flamingo, the migration birds, very famous. So these birds fly from the one end of the world to other end, and they go during a particular season in Tanzania, and they feed on that lake because those lake has a lot of carotenoids, like microbial carotenoids, which are consumed by that. And that's why the reason of the flamingo also they appear orange or red color. So even we see the lake, it is orange or red color. And some time of the season, it's completely red, that is full of carotenoids in that lake. So it's a natural process. There's an interaction which is happening between algae microbial consortium, which is producing those microbial carotenoids. And thus, you can see that by observing the environmental conditions, you can also design your work to naturally understand what is happening between that. So what is the rationale behind the work? So we are trying to understand the physiology of algae and the bacterial interactions and illustrating the fundamental metabolic process in producing the high value products using omics paradigm. As I said, we don't have the sequence of this particular microalgae. So we went for a feedback omics approach where we try to understand applying the phenomena of metabolomics to reveal what is happening at the cellular metabolic functioning pathway, which gives a blueprint for the bio-based economy or a green manufacturing. Because biomanufacturing is the need of the hour, where we can produce indigenously many products from the natural strains. Thus, we have to capture and understand what these strains are able to produce so that these products can be manufactured using non-GMO approaches. So I'll take a little bit time because this is very important in terms of methodology in order to understand. And also because I know most of the participants are students, and also we have some faculty here where we have to understand how to design an experiment in order to understand what is very important, how to capture the overall profiles of those systems, what we are looking upon. So you can see there is a term called balance ratios. So it's very important that when so I'll before going to balance ratio, I'll just take a minute to tell why we have to do this work, because what happened is we always maintain our algal what you call algal strains, because you have to recycle your algal strains on a regular basis in the lab, and we maintain our parent cultures in different agar media or what tap media, all those things. So basically, when we are trying to discard a few of our strains because those have been older, and we have seen that my student have observed that, sir, there is some problem in this, and I can see some algae is there still, but there are some orange spots on the algae. I'm interested to look upon what is this. So when we were about to discard those plates, but we thought, like, let us see what is this. So basically, when we found an orange spot, we thought algae is because it is almost 20 days or 3 months, algae is producing carotenoids, and it may be algae itself as a separate spot. So we never thought, because these were axenic cultures. Okay. So what happens is, even we maintain axenic cultures, some of the bacteria are present with that, where they are very beneficial. If you try to remove that bacteria, the growth of the algae can come down. Okay. So there are certain experiments which have proved, some certain researchers have also shown that the beneficial bacteria, if you try to remove it, they may not be. So in that way, by accident, by this thing, we came up with a consortium where we thought we took that bacteria We made it single colonies of that bacteria. At that point, we also don't know that whether it's a contamination or something even beneficial or not. So what we have done is we have sequenced that bacteria, we have done 16S, and we have sequenced that bacteria, and we have seen that it is named as Exicobacteria. This is very unique in the sense Exicobacteria is an orange yellow pigment producing bacteria, and that orange color is nothing but the phytols and the vitamin E, vitamin components which they can produce, and that gives color to that. So that's how we came up with this accidental bacteria. Now we started designing our experiments because in that media where we are growing the plates, only algae can grow. Okay, bacteria cannot grow in that media. So if a bacteria can grow, that is in consortium with that, it should be a symbiotic bacteria. So we now started to understand. Now we thought like let us start working, adding 1% of this 1%, that is 1:1. So 1 cell of bacteria, 1 cell of algae. So we started different ratios, and this was a very huge challenge for us because we tried to manipulate the algal content, algal cell number, and also bacterial cell number, which has been very difficult for us to come to a conclusion what is the best option to get better growth of algae. At this point, we were interested that algae only should grow along with the bacteria because the media, the dominance should be by the algae, not by the bacteria, because the media of what we are growing, bacteria can grow, but it cannot dominate the algae. So we started designing a concept called balance ratios. What is balance ratio? So we keep one organism constant, that is algae constant. That means we take 1 cell of algae, 1 cell of bacteria. Now change the composition of bacteria like 1 cell, 10 cells, 100 cells, 10,000 cells, and 10 to the power of 5 cells. Like that, we try to change the bacterial population, not the algal population, and develop a concept called balance ratio, where we have seen highest growth in 1 cell of algae and 1,000 cells of bacteria. That means your bacteria is growing, but the algae is still dominating the growth of that. So that ratio is called optimal or balance ratio. And this is very important because all the algae, all the bacteria cannot grow in a same phase. So you have to understand how you can design your experiment so that you have certain algae composition and certain bacterial combination with the dominance of algae, and algae can grow happily. So that is how we try to capture that 1:1,000 ratio of bacteria, and thus balance ratio was used for all our experiments further in order to understand the metabolomic experiment. So you can see we have some basic experiments what we do upon. So now we add 1 cell of algae and 1:1,000 cells of bacteria so that we can quantify through confocal microscopy what kind of lipids or what is it producing, antioxidant activity, all these things I'll be discussing now. And through GC-MS metabolome, we try to analyze what kind of metabolites it is producing in order to show interactions of those things, and HPLC in order to understand that. And friends, this algae is a very unique algae which can produce lutein as a major product. Okay, as I said, all the algae can produce high value product, and each one has a different relevance. The product what I was talking about, transcriptional engineering, is a product which is called astaxanthin, but this algae can produce only lutein, which has a major constituent of carotenoid. So we try to keep it as a benchmark where we can produce lipids, where we can produce lutein as a major high value product without compromising the growth. So this is the overall concept how we design the balance ratio of axinic bacteria and axinic bacteria and axinic microalgae together in the consortium so that it can produce a high value product like lutein, vilozanthine, anthrazanthin, or beta carotene. These are all high value products, but the end product, the major product, what we are getting is lutein. How we are getting it, we'll understand in the next few slides. So this is how you can capture the basic parameters of a system, as I was explaining in the previous slide, like how you can utilize different media composition of phototrophy, heterotrophy, or mixotrophy in order to produce the high value product of interest. Here, when bacteria and algae are working together, they are able to produce lutein as a high value product. We are also working in depth with the bacterial analysis, how bacteria is getting benefited. And we have seen that bacteria is able to produce high quantity of vitamin, which is very unique, where vitamins cannot be produced by microalgae, and microalgae requires vitamins for their growth and other processes. Thus, they are and the products which are being produced by microalgae, such as glucose, other high value products, as is like some of the things which we'll be discussing, they are exchanged with bacteria, and bacteria is producing different products. So we're also looking simultaneously bacteria and microalgae physiology in order to understand what is happening to the metabolic pathways. But today we are going to just talk about how the interactions, because this is how anybody anywhere in the world can plan a simple experiment in order to design the work and get a high output data in order to understand the overall process of the how the interactions can work upon those things. That's why I'm trying to more emphasis upon how you can design your experiment, how you can focus of producing this. This is again a non-GMO approach, nothing to modify, just fine tune your media engineering compositions, media by understanding the balance ratios, and try to do the basic experiments and do a classical metabolome to all. How do we understand those things will be discussed in later slides. So as I told in that, this is the first result. This is how our slides, or what we call slants, looked upon. So this is about to, we tried to about to discard these. We can see orange and yellow color things. We thought it's a contamination, let us discard. But my student was more enthusiastic. Why can't we look upon this key? What kind of associations? If it is algae, that's fine. We can look upon the different concept. But we thought like it should be some contamination which is bothering us. Still, you can see that algae is growing happily. Even the bacteria is there. So what we have done is we have isolated this orange or yellow spots from the slant, and we went for sequencing, and we have seen that when we see that this is very much close to Exigobacteria species. And there are two types of Exigobacteria: cold adopted Exigobacteria, where they can work in cold adaptation conditions. But what we got is a hot adopted Exigobacteria, which ranges from 15 degrees to 40 degrees, and it can withstand high levels of pH. So it is very interesting, and it's very and we have certain forms of Exigobacteria, and that's how we try to understand what basically giving the color for Exigobacteria. And we have quantified from our bacteria species, and we have seen that this is vitamin. Because of that, there is a change in the color of that. So you can see the Exigobacteria are present everywhere. You can see in water slides, they got in effluent, they can be found in fish, this thing. So it's a basic bacteria which is present, but it can be hot adopted and cold adopted Exigobacteria. Microalgae we grew at 22 and 24 degrees centigrade. So this bacteria can also live happily, and it has a broader range of pH and temperature. So it can easily go up with the challenges what is upon that. The basic surprise is whatever media microalgae we are giving, the bacteria also can grow. So it has adopted itself to that, so that it can. So we have used Bacillus indicus as an outgroup so that we can get the phylogenomic. It is very important to put an outgroup also in order to understand whether we are doing our phylogenomic analysis correct or not. It's very important in that point. So coming to the second perspective, second experiment, or a first experiment, balance ratios. So I spoke to you guys about the balance ratio, where we try to use different ratios of 1:1 of A is algae and B is bacteria. One cell of algae, one cell of bacteria together, and you can see the specific growth rate and doubling time. You can see the biomass productivity goes up high in 1:1,000 cells, which is the considered point. You can see that this is algae alone, and this line on the top is algae plus bacteria in 1:1,000 ratio. So it has increased a lot, and the biomass productivity has been increased a lot. One thing what we have kept is usually algae grows at 100 Einstein, which is roughly good for the algal growth. But because we had bacteria, initial experiments we have done at 50 micro Einstein, so that light is also captured by algae and bacteria also grows happily. Now we have improved this variety. Now we can grow at 150 micro Einstein, no problem. Bacteria also can grow up. So we have adopted both the consortium to grow at 150 for best suitable for microalgae, so that we can do further experiments. So in this experiment, we have not used carbon dioxide, we have not used any light effect, but only we have seen that how these two are interacting to each other in order to promote the growth or without compromising the growth. It's always better than single algae. It's almost doubled 1.1 fold increase or 2 times increase in the production biomass productivity, which is very good in terms of microalgae. So thus we try to obtain a good high productivity, keeping the ratio. Balance ratios are very important. So that's how we try to prove. And this experiment alone took us 3 to 4 months to standardize, because it was very difficult initially. We were using different combinations of algae, different combinations of bacteria, as I said, which was very difficult for us to handle at a huge. So the combination was becoming huge and huge. So that's how we came up with this balance ratio, and we were successful in demonstrating that there's no compromise, or there is enhanced growth productivity, biomass productivity of algae, rather than nothing to do with the bacteria. So we have bacterial growth also, but I'm not showing because, as I said, we were interested to understand what is algae doing for us. As there is a separate study where we work on bacteria alone, and we also we compare those things and also do that. So basically, these bar diagrams are the base constraints. So we do all the experiments in a time course. What is a time course? So in order to understand the omics experiment, it is very important to demonstrate your change of expression of any protein or gene or a metabolite with respect to hour or time points. That's called a time course. For example, we have done the experiments in 0, 3, 6, 9 days in order to understand how our lutein productivity is enhanced without compromising any growth. So on our bottom, I can show that the total pigments, that is carotenoids, chlorophyll B and chlorophyll A, enhances huge. So the plus point of our study here, we are not using anything other than bacteria, and we can see that the chlorophyll A is enhancing huge. That means that the growth profile or biomass productivity is enhancing also. That is a positive indicator. Along with because algae is growing, the carotenoids, the carbon flux is also going towards carotenoids. That is also one factor where the carotenoids are increasing. Basically, this is also lipid producing bacteria, but we can see that lutein is a major product which has been produced in that. So you can see other products like lutein. So when we quantify different carotenoids, we see that there is a production of viloxanthin, anthocyanin, lutein, and beta carotene. Now, when we are scaling up, we are even getting zeaxanthin. So, friends, if you are familiar with the MEP pathway, which is very popular pathway for production of carotenoids, and it's only one pathway is present in microalgae, that is MEP pathway. Whereas in plants, MEP and MBA pathway, both pathways are present in that. So by using this MEP pathway, there are two major constituents, that is beta carotene and alpha carotene. Through alpha carotene, you will produce zeaxanthin. So beta carotene, you produce lutein. That is why the beta carotene component goes down and lutein In some of the products, like some of the strains like Duna, beta carotene is a major product. So lutein is not produced in that. So every strain, based on the environmental conditions, they developed a method to produce a carotenoid of their relevance. That's Dunarilla at particular stage will always be seen as red, but lutein we see later as an orange or reddish yellowish tinge. But beta carotene, it's always red when it is produced in excess. But beta carotene is converted into lutein. That's the reason why we are not able to see more of beta carotene. Thus, you can see that there are different molecules which are being enhanced in a time course experiment. It is very important because all the omics should be done with respect to time, because relative profile enhancement happens with respect to time, not with the parallel control. So this is very important parameter which has to be followed upon. So this is how the methodology we have worked upon. As I said, we used algal and bacterial inoculums, and we have different conditions. Initially, what we have done is we tried to look upon different conditions where we used algae and bacteria in that concentration, and we also subjected to depletion. So in order, because we know that when you remove nitrogen from this media, it can produce high value products like carotenoids because it is in the presence of light. So we try to apply that condition where can we enhance the production of lipids or not. So we have algae alone, algae bacteria consortium, where we have given nitrogen deplete as one of the condition. And we have another thing, nitrogen replete where plus CO2, where carbon dioxide is sequestered, and that carbon goes towards either carotenoids or what products we have tried to understand, where we try to see that media, it changes to, it be converted into, sorry for this is NR condition plus CO2, where we are substituted in this condition only carbon dioxide so that it can produce high value products. So again, we have done the cultivation for 9 days, 0, 3, 6, 9, and we have seen that both biomass and carotenoids have enhanced in the presence of bacteria. That is what we are expecting upon. So these are the different kind of media engineering approaches, so that when we are scaling up this technology, we can scale it up based on the information what we get from the basic lab experiments. So in that also, when we add, so no carbon is NC, HC is high carbon. We have kept the ratio constant. You can see that the algae alone itself is so green, but the consortium is much greener. You can see from my biomass concentration where it has enhanced much with CO2. Without CO2, the previous growth biomass productivity has shown that it is around 546 biomass productivity mg per liter per day, but with carbon dioxide is almost 1 gram per liter per day. So that means it is again doubling to that core, and we can see that even there is a slight change in the pH, but that pH is fine for both bacteria and algae to grow upon. So we do different parameters because we are substituting the pH. When we are substituting with carbon, it comes between 6 to 7, so that the algae can happily grow upon. Where there is no carbon, it can be more higher than that. So basically, when we started, you can see that algae alone itself, that leads to some alkalinity where it is around 9 to 10 pH, depending on what kind of algae you are working upon. Especially, you need a pH parameter if especially we're producing a carotenoid producing microalgae. That's very important. These are the basic conditions we need to understand when we are working with algal systems. This is again the carotenoid profile. You can see chlorophyll still goes beyond that, and carotenoids also enhanced. And we have seen that when there is a carbon flux, now additional carbon source, that is carbon dioxide supplementation, we have seen that viloxanthin, zeaxanthin, and lutein have been enhanced multifold along with that. So now we are scaling up at a 5 liter and 10 liter biophotoreactor, where we can understand how do we produce, because lutein and zeaxanthin is a highly important commercial product which is directly used as a nutrient supplement for the people who have problem with the eyesight. Okay, because lutein and zeaxanthin are the two complements which will help these carotenoids will help to streamline the light which is falling on our retina. So it's very important. And if you go to a doctor or any eye specialist nowadays, they are giving a supplement which has a composition of lutein and zeaxanthin, and this is a naturally producing lutein and zeaxanthin strain. So we can see that both beta and alpha carotene are at lower levels because they are converted into lutein and zeaxanthin. So either you can use the combination of that, or we can use the produce. So now, in order to only produce lutein, we have to shut down the beta carotene pathway so that lutein can be enhanced, or we have to because as I said, pharmaceutical companies are okay with the composition of carotenoids with the combination of lutein and zeaxanthin. They can directly use in their formulations for using the nutraceutical products. So that's the combination. So when we are supplementing, there is a flux distribution between alpha and beta carotene. That's why we can see lutein and zeaxanthin together. So these are very good parameters where we look upon. So when we have scaled up and we have introduced carbon supplementation, so there is a certain threshold where the organism can produce this much amount, and then it starts distributing to other components. So that's how it is working upon, and we are deep diving using our transcriptomics and proteomics how this is behaving. And there is a reason why zeaxanthin and lutein interplay together, because zeaxanthin cycle plays a viloxanthin and zeaxanthin cycle plays a very important role. That's why there is a role of viloxanthin and zeaxanthin also when we are scaling up and there is excess of carbon. So this is how we got a unique finding that you can produce a combination of carotenoids when you can give it an excess of carbon. So this is how we do with HPLC profiles. And you can see this is what I'm talking about. You have alpha carotene, which is producing lutein, and beta carotene, which is producing zeaxanthin. This is how the pathways look upon. And you can see different combinations. You can see the peaks changing in terms of 9th day, where you have high lutein content along with zeaxanthin and this thing. So thus you have these HPLC profiles where we look upon and quantify through standards and to understand how these different metabolites. And this is a very well-versed method now in our lab, and you can follow our protocol in order to extract this. This is a simple protocol you can do and quantify different carotenoids from that. This is so now comes the metabolomics. So as I said, like we have went through an approach called a reverse omics approach and identified different. So this is basically a PCA plots. We represent microalgae alone, bacteria alone in B and C co-cultures. And you can see 0, 3, and 6, 9 form in a same cluster. That means these are biological triplicates, and this information lay down on each other so that we can see that whether our conditions are working together or not. And PCA analysis will also give cluster analysis. Will identify the metabolites, because you have to do a biological triplicates with technical replicates in order to overlay and see that how your metabolites are being identified in the time course experiments. So once we have the metabolites, we convert using metaboloanalyst. So this is a software which I'll not be taking because it itself takes a lot of time to explain, but it's a very easy to handle software, user-friendly software. So basically all the raw data, the HPLC and GC profiles which are created, you can download those as files into your metaboloanalyst software, where you can convert and identify those metabolites, and also convert these values, whatever you are quantifying using the GCMS, into different heat maps. And these heat maps give you how different metabolites are getting converted. And major classes of metabolites, what you can see is amino acids, fatty acids, organic acids, sugars, and alcohols, along with other vitamins and all those things which can be. And you can see that Basically, algae and bacteria alone have around 30 to 40 metabolites around, but the interaction showed more unique kind of metabolites which are absent and also present along with that. And this is a consortium which we are trying to understand upon, and same class of amino acids and fatty acids along with other things play a major role in the consortium studies, and how these both are being exchanged between together to in order to roll upon those things. That is how. So these are the list of metabolites. So this data is recently published, and if anybody wants, I can share the paper. It's a this is the metabolites what we found out. These 48 metabolites are the metabolites which have been major metabolites, because we put a threshold for the minor metabolites we don't look upon at this point. So we developed a LC method where we look upon those and map on those other metabolites of interest. So for example, some of the metabolites also we lose upon, but these are the major constituent of metabolites what we found, like amino acids or lipids or all other things in turn. And you can see on to my right, this is called fold change of between two conditions, and describing between 6th and 9th day, where you can see that upper part is upregulation and down part is down regulation. So you can see the pink dots which are available, or purple dots, which says that these are upregulated metabolites, and 2 and 4 are the threshold or fold change. We consider most of the metabolites which are upregulated and downregulated. So some metabolites get upregulated and some get download. Anything lesser than one fold change, we just do not consider. Maybe this is a transient expression, but is basal level expression. But these are not very relevant in terms of what we are looking upon, or this the basal level expression will always be there. So remember, any omics data, we always have a threshold where the basal level expression is nullified, or we don't consider at a certain fold change level, because that creates a lot of biasness. So in order to reduce the biasness, we consider, we put a threshold of 1 fold or 2 fold depending upon what you're looking upon. And that's how we try to understand what are the metabolites which are getting downregulated and upregulated in the scenario of this. Based on this data, we come up with this schematic outline or overview. Thus, we can reconstruct the whole pathways, and you can see this is Exicobacteria and this is the algae, how the exchange of different products based on whatever metabolites we have produced or we have gotten have been involved in converting these metabolites into different. So I'll take a minute here in order to how, so I've told, like, we are trying to increase the lutein as a major product because this is the carotenoid or lutein producing bacteria, microalgae, and how bacterial compounds are being exchanged with algae in order to look upon how they are growing up. So as I said, we in this paper, we were not discussing about anything, how the bacterial component is working upon, but it is very important that we are looking upon how bacteria is also in symbiosis getting benefited from the algal products which is producing. So you can see that bacteria is also producing a little bit of CO2, also bicarbonate in form of sugars, which can be used, and it can feed into Rubisco chain Calvin cycle, where it can fix this in form of a glucose glycerol 3 phosphate, which enters the MEP pathway, and thus it can enhance the production of lutein and all. MEP, the G3P or not only enters into MEP pathway, it also enters into central metabolic pathway, and pyruvate also comes into GPT to produce that. So basically it is using different compositions. These are the factors, and as I told initially, vitamins are very important for microalgae, and microalgae cannot produce vitamins by itself. So we have a vitamin complex which is being produced by bacteria, which is coming into the media, and this is getting utilized and entering the central metabolic pathway of the microalgae. Same wise, amino acids and some sugars are entering the central metabolic carbon pathways, so that it can go up to TCA cycle and also involved with the biomass production. Thus, your biomass is either enhanced or not compromised under the unique production, or there are some heteroaromatic compounds which we have discussed earlier also, where these products are involved with feeding them directly into TCA cycle. When you have a active glycolysis and TCA cycle, that means you have an active um, production of biomass. That means when the biomass is increasing, the remaining carbon is going towards the lutein or viloxanthin or astaxanthin, what we are talking about, which is able to produce these compounds of interest. Also, the production of carotenoids can scavenge the ROS molecules, which are very important. ROS which will be generated in any kind of stress. Here, why we are talking about ROS? Because we have given a condition of a nitrogen deplete condition where ROS molecules can be scavenged. That's the reason why we are subjected to nitrogen depletion, so that the stress can produce more carotenoids, and those carotenoids can scavenge the ROS, and the algae can live happily. So most of the factors which are required by the microalgae are given by bacteria, and those are fed into the central metabolic carbon metabolism, either through these into TCA cycle, and are leading to biomass synthesis, or the carbon dioxide or bicarbonate which is present is coming to the Calvin cycle, and the carbon flux, as I said, like you have the more carbon available, the carbon is fixed in the form of carotenoids. So it also not only forms the carotenoids, it also forms other sugars. We have observed that there are multiple sugars which are being produced in this, and these sugars are also important for microalgal growth. So same wise, pentose phosphate pathway, we have seen that there is a reduction. As I said, like we have the initial studies gave the leads that how the interactions are behaving, and we have identified certain markers or compounds which are being produced by bacteria alone, because these are not seen in the studies. These metabolites are not seen in the co-culture experiments where the growth has been reduced or decreased lesser than the co-culture studies. So thus you can utilize the environmental factors where the exometabolites can be shared between the two strains for their either the growth, and that's the coexistence will help us in the production of any relevant high value product of our interest, or thus it is very important, and thus it can be used in producing multiple products. This is one example how the basic things can be understood in the environment. And the microalgae, what we are talking about is chlorophyll, chlorella sacrophylla. This is a sacrophylla, and the bacteria, what we identified is Excicobacteria. So there are a few studies like Excilia helleri, the first slide, what I was studying I was showed as a case study, is also one of the example, well-studied example. And they have also shown that this exchange of metabolites may either lead to coexistence of microbial algal interactions to produce certain products, or they may use to pathogenicity depending on what they are producing upon. The conditions prevail, and you can control the conditions, and these conditions are very cost effective so that you can mimic into a high scale level in order to produce. Now what we are trying to do is we are scaling up these experiments in order to understand what is happening to the carbon flux, and for example, that technology can be used to efficiently observe the flue gas concentrations or lesser than flue gas concentrations, so that carbon which is a waste can be fixed into some high value products. So that's the concept of waste to wealth can also be there. And we can also use the industrial wastewater effluents or any of these to grow. We don't require anything. So these experiments can be done. But because we are talking about high value products, most of these experiments should be done in controlled conditions, so that even it is a non-GMO, because we have to do in a controlled conditions in order to produce high value products. If the organism is going to consume directly as a nutrient product, then it should be close to this thing. If you are extracting the metabolites of your interest and using that will increase the cost of processing, post-processing or pre-processing. In that way, it will lead to little bit cost constraints, and you can grow in open ponds also. But most of when you talk about carotenoids or high value products, these should be closed containers, either it is GMO or non-GMO, because there are certain rules and regulations which has to be followed upon in order to do so. But in that way, you can capture, so you can use this, and you can also employ. So most of the things what we are trying to do is we have to extract this compound so that the purified compound is always having a higher value. This is how, finally coming to a little bit of conclusion, how we have worked upon. We have identified a new symbiotic bacteria, Excicobacteria, which has a biotic relationship with the microalgae Chlorella sacrophylla that enhances carotenoids, especially because I told like this is a lutein producing bacteria, and we have shown that there is a balance ratio adaptation of the experiment where we have seen that there is an enhanced growth nearly when compared to the other conditions, and our so that vital role in carbon metabolism and associated with other pathways using a reverse omics approach. Under 3%, we have also seen that there is an increase in productivity, lutein productivity to almost threefold change, which is a good in that approach where we are trying to understand. And also we have seen that there is a deepoxidation of molecules with energy dissipation capacity. I'll not discuss this thing in detail because these are the scavenging molecules or carotenoids which are involved with that energy dissipation capacity. And we have done some PAM studies to understand this. And this is very important to understand how your lipid is scavenging, carbohydrates are scavenging the energy which is present in it. As I said, the violozanthine cycle changes the photosynthetic pigment levels, especially B and beta carotene. And we are looking deeper into this. And now we have done, we have certain leads now that I'm not discussing because those we are under development stage where we are trying to link the overall process where violozanthine cycle is involved. That's the reason where we are trying to understand that violozanthine cycle is a main component which is playing a crucial role in enhancing the carotenoid contents. In conclusion, this is a basic understanding of violozanthine in algae. Not much is known about that, especially in algae, but this is well adopted in plant systems. So basically what we confirm that whatever algal systems which are more primitive systems have these metabolic pathways have been passed on to the next highly multicellular evolved organisms such as plants and all. And we understanding these basic biology of how these metabolic pathways are being regulated will help us to understand how different pathways are being regulated in other systems. Thus, this kind of scenario will help us to understand how different pathways can be regulated and without modifying. Especially, we need an approach called a non-GMO approach, where you can do it in the open environment or closed environment. You don't require much of regulations in order to understand these things, and you can happily grow relevant high value products. So using the environmental constraints or capabilities of that microalgae, it is very important how your cells are behaving, and that can be translated into the value product what we are looking upon. So this concept of reverse metabolomics will help a new enlightenment to all our participants. So you can think in this direction also, because doing genome sequencing is a tedious process and it may end up with a lot of experiments to be done and you need an expertise. But this is also a concept to basically come up with a new way of understanding where you can, because once you understand the metabolic exchange between the coexistence of the algae and bacteria, it is very easy to work upon the product of your interest. So that is a concept of coexistence we brought into a new this thing, and this paper has been published, the basic data. So I'm happy to share if this paper is required by any of the participants. As I said, like you can email me or anything, and I'm open for any collaborations if anybody is interested, especially who are working on this kinds of concept and we are interested, because our lab works on basic biology of multiomics, and we can definitely collaborate in this consortium in that way. So finally, my special thanks to all my international and national collaborators. We worked on different projects. For example, I have not discussed on omega-3 fatty acids, heterotrophic growth. We have worked on other products like fucoxanthin, tocopherols. That project is also not being explained in this. So we have, as I showed in my roadmap, and these are the papers or publications in our lab since last 3-4 years. And most of them are available online. And if anybody is interested, you can email me and I'm happy to share that. And last, I would thank our funding agencies, which keep us constant and given us a good opportunity to explore these areas of research, which is very important in terms of exploring the sustainable trade for the production of bioresource, which is the need of the hour. And we know that biomanufacturing is the way where we can strengthen our economy, not only exploring the wide varieties of algal species what we have. And this is a simple way where you can uniquely produce and you can scale it up the technology without doing any high level modifications or overexpression of certain genes. But this is very important to capture the novelty of the cells and how they are behaving at different levels. So thus, it is very important to understand the basic biology so that we can translate that basic biology into a product of interest. And last, I would like to thank Dr. Nas and Dr. Tiwari, especially for inviting me for this talk. And also, it's a great opportunity for me to share the ongoing research in our lab. And also, I'm happy, as I said, I'm happy to collaborate or interact with you, and you're open to come up. I shared my email also, and it's always a pleasure to work with multiple PIs and investigators, so that will help us in exploring more and more ways of avenues, so that we can together build a better way of understanding the overall algal research. Thank you.
Thank you, sir. Thank you so much, sir. Really a wide knowledge you have shared with us and new to us. Thank you, sir.
Yeah.
There are few queries, sir.
Okay.
So the first one is from Parmeshwari. Sir, can we use algae to produce bio battery or fuel cells?
Yes, that also is one of the area which is still working upon. So we try to collaborate with one of the other institutes in working on little bit work has been done, but still this is a very niche area. So it is very relevant that the combination of physical and chemical fuel cells is the way. How do you make that algal bioreactors? Because solar energy can be captured by this, and there is a process where you can use in the what you call reactors for the algal powder in order to capture that. So this is a very, very niche area, and this is always a possible way, and this is an alternative for some of the solar cassettes what you are talking about. Yeah.
Second query from Sony Sigma. When we grow microalgae and bacteria, how can we separate them during purification?
Oh, that is very easy. It's a pre-processed technology, and lutein is being produced in large amounts from the plant sources, especially sunflower. And this process is already developed. So you can segregate lutein. It's through normal LC-based approach, and the pre-processing also it's very well established in the industries. So lutein is already produced. Only thing is we need different feedstocks, and because plant is highly competitive in the sense like algae can grow very faster than plants. This is an alternative source of producing the lutein. So you can purify the lutein from the mixture. It is not at all difficult. Yeah.
Another question from Parmeshwari. Sir, can we use the synergistic growth of algae and bacteria to synthesize nanoparticles?
Yeah. Nanoparticles, you can use algae alone itself, but what does bacteria help to synthesize nanoparticles is a study to understand. Okay, so in a consortium, Usually algal nanoparticles are well known, and you can also know that algae can be used as a nanoparticles for some of the things for production of nanoparticles. But why we need to have algal and bacterial consortium, that is very important. So how you are going to use that consortium to use that product is very important. Okay.
Sir, many appreciations from the participants, sir. Very satisfied with your excellent presentation. Thank you so much, sir. Raju Ganeshan, sir, do you have any queries? Can you unmute?
Yes, ma'am. Yeah,
Yes.
hello. Is it
Yes.
voice audible, ma'am? Can I
Yes,
ask?
yes.
Sir, first of all, thanks for your presentation, which was so excellent. So recent years, I'm working in metabolomics. So I have some small questions. When you are doing the peak intensity in GCMS, how you are converting that peak intensity to the score plots? To the PC,
Okay.
you are analyzed the PCA analysis and PLSDA and OPLSDA. So do you have used any of computational tools to convert the intensities to the Excel values, and while you are analyzing to go for the metabo analyzed, I hope that software is you used. So from the Dr. Vijat groups. So so what is the tools which was how you are converting the intensity GCMS intensity values to the score plot?
Okay. So that's a very good question and a technical question. So it's very interesting. So what we do is we have an approach called internal standard and external standard. So we use the non-productive product as internal standard. For example, all these experiments we add ribitol, right, for internal quantification. Also, external standard where we try to run different standards based on the initial profile what we get. For example, we make different quantitative values, standards, quantitative graphs of those standards, which are available in Sigma and all. So we also run those as external standards with the same method, and we have a peak area. That's how you quantify the internal this thing. So whatever area intensity is done, when you have a particular file which has the peak area intensities and all, so you directly transfer that file into MetaboAnalyst. That will convert your peak intensities into a quantifiable value, and those can be converted directly. So when you go to a MetaboAnalyst, it will give it, and that's why this method is called semi-quantitative method, where we can approximately convert the values of peak area into that. But exactly what it has been produced, we convert that with the external standard. For example, now I am looking for lutein. So I run different standards of lutein externally. I know the peak intensity of the lutein and directly convert, just like in biochemical, I convert the individual lutein into that. But when we are doing at a global metabolome level, we try to overlay the peak areas, and that peak areas values are taken it up, and that is converted in the MetaboAnalyst as those analyses are done in the form of heat map and all those things. So those fine tunings are done manually, and this is a very good, interesting. So next month we are conducting a metabolomics workshop for in ICGB. Yeah. So in that we are going to cover these things. Yeah.
I would like to participate. So another one question, Professor. So when the PCA score plot, when I was continuously working in the x-axis, the PC principal component, one value, the percentage wise is very less, which means below 4 or 5, below 10, like that. But the y-axis, the principal component PC2 values as more. So if it is 30, 40, 50. So I cannot differentiate what is the score plot interfering the meanings from the data, from our data, what it is interfering, what it is tells the meaningful differentiation. So according to this score plot, the principal components values.
Yes, basically, values. Yeah. So basically, the PCA plots will help us whether it's laying within that umbrella or window what we are talking about. So for example, each biological replicate, that's why it is very important. When we do once, all the metabolites will be at random. So if we can put the same thing I showed at a conditional level, but if I take it to metabolite level and put the PCA plot, it based on your expression value of that metabolite, your PCA variation will shown upon. So for example, one condition A, that is a biological replicate A, and biological replicate C is showing a huge difference in your metabolite, then you cannot consider that metabolite expression profile in that experiment. So you have to replicate those. So the basic technique, what we do here is, we do each sample in 3 technical replicates and also 3 biological replicate in order to reduce those kind of hindrances. So when you reduce, it will overlap almost, and that 5% error rate can be taken into consideration, but not, as you said, like there is a huge variation. So it is an experimental, what do you call, manual experimental error which happens while extracting process. So there is nothing to do with that one. If it is a condition 0, it will be the same metabolite profile, but condition 9, it may go up and down. So for example, I am comparing a metabolite. In this case, metabolite A in condition time 0 is at some 10 or something, but on 9th day it is going to 40. So my PCA plot shows that it is sitting somewhere. So that's how initially, that's why we try to overlay each time point and each metabolite, so that you can consolidate and look upon. So it's very tricky when we are doing global metabolomics. We may end up with lot of questions and overlaying patterns. So whatever outliers are coming, you try to remove those outliers. And when we have multiple technical and biological replicate, this problem can be reduced to less than 1%.
And Professor, I have one more question. So the Kenomics software, so for which is using for the proton NMRs based metabolomics that is a Kenomics and Shimka for the score plot analysis. How does this software is working? So I want to learn those things. Do you have any kind of those kind of tools and software for metabolomics in specific?
Okay. So basically, we do LC and GC metabolomics. We don't have NMR at this point of view, and NMR we have challenges. So we want to do global metabolomics. We cannot do at NMR at this stage, because we have to have very high concentrated sample in order to understand those things. So NMR, once we get the metabolites from the NMR, that can be overlaid on the KEGG mapper to understand what kind of metabolites are getting. But at this point, we combine GC and LC metabolome to overall understand the global profile of that. So they are different, because basically these are all conversion softwares where you can understand the machine language to the molecule. Also you can use these peak areas in order to extrapolate your data, what you're giving. Most of them are semi-quantitative, but the correct way of doing quantitative analysis, you should always quantify external standard and also compare with that. So otherwise, you are not going to get an accurate value.
And one more last question is, don't forget to invite me for the metabolomics workshop. I would like
to
join.
At this point, thank you very much for the interest. But at this point, we have closed this thing. But you will be getting, so we will be doing those workshops every year. So definitely you can apply on that. But because that date has been gone 5-6 months ago, because usually we open 1 year before for these things. So I will share.
Thank you so much. Thank you so much for your nice lecture.
Thank you. Thank you. Any more queries from the participants? Sir, one more question from Jyotishri. Can we encapsulate the microalgae or bacteria in any polymer beads and grow them for better downstream process?
Yes, it is possible, but that depends on what downstream process you are looking upon. Why you want to encapsulate microalgae or bacteria? So whether you want to work on just a carbon sequestration component, or what is the downstream process you are looking upon? So for example, if I want to do a biofuel extraction, I don't require to put in polymer beads. because microalgae grows up. I put in a hydrothermal liquefaction. I get the crude lipid, I distillate it, and I get the biodiesel. So it all depends on what is your thematic. So polymer beads or polymer approach is being done in order to what we call it as you make them cells available there itself, and you can do a sequester kind of thing. So it all depends on what kind of downstream approach you want to do, what is the advantage of that. And remember, when you're doing high value product, you can add certain cost to downstream process, because high value product, especially like lutein, astaxanthin, 1 gram cost in more than $1,000, right? But biofuel, you don't have that option. Biofuel should be cheaper, economical, and more quantity. So you have to understand, when you are trying to increase the downstream process, it will increase the cost. So it's the best, quicker way of method is produce the lipids, do the HTL process, and that's the only way now available with whatever technology in the world. Supercritical extraction can be used. Again, it depends on what kind of product you want to understand. So supercritical can keep your product pure in terms of carotenoids and all, but in terms of lipids, HTL is workable solution. So it all depends on why you want to capture it, because you're adding cost to that. And if you're, as I said, you can use the microalgae directly as a supplement for the food also, nutrient supplement. So, but you should be careful. So when you are talking about bacteria and algal, so we are using the natural phenomena. As I said, that it's happening in the environment. We are not creating something new. It's happening in environment, but we are using the capabilities of microalgae to enhance more in easier and quicker way, because no industry will wait for 20 days to produce their product. They want everything quickly in 1 week or 5 days, because you are reducing the cost. 20 days into it will increase the cost. That's where we are trying to understand, increase the biomass, reducing the doubling time. These are the different basic approaches where we can understand a lot. There are different methods. As I said, it all depends on what is your end product and what is so we are more interested towards biomanufacturing of natural products, which is a need of an hour and which has huge economic importance, and also look upon alternative sources, keeping in different scenarios like carbon sequestration, using wastewater, seawater in order to grow our microalgae. Yeah.
Thank you so much, Pawan sir. It's really a very enlightening talk today. So, and I would like to thank Tiwari sir also for giving us such a good platform. On behalf of our organizing team, we are once again thanking you, sir.
Oh, thank you very much. Thank you for inviting me, and it's a pleasure to share our research with all who are interested, and hope I could even introduce what we are doing upon, and looking forward for any kind of collaborations. Thank you.
Thank you, sir.
Thank you, sir. It is our privilege to hear your expertise and insights that have greatly enriched our program and provided valuable guidance to us. Your enlightening lecture during our online session has made a lasting impact on all of us, and we're immensely thankful for the opportunity to learn from you. Thank you everyone who had joined and acquired knowledge from today's session. Kindly join tomorrow's session at 10:15 a.m., followed by the valedictory.
Thank you.